Today I have Kristin Sainani. Kristin Sainani is an associate professor at the Stanford University. She teaches statistics and writing, works on statistical projects in sports medicine, and writes about health, science, and statistics for a range of audiences. She authored the health column Body News for Allure magazine for a decade. She is also the statistical editor for the journal Physical Medicine and Rehabilitation, and she authors a statistics column, Statistically Speaking, for this journal. She teaches the popular, massive, open online course, Writing in the Sciences, on Coursera. And she was a recipient of the 2018 Biosciences Award for Excellence in Graduate Teaching at Stanford University. So thank you all for being here, and I will now pass it over to Kristin for our presentation today.
Thanks, Jackie. So yeah, today I'm going to be talking about how to analyze research data. And my goal for today is to allow you to peer under the hood of a real data analysis. Unfortunately, I think many PhD students and applied researchers are never taught the data analysis pipeline. They may be taught just, a tiny sliver of data analysis, such as, how to run specific statistical tests. But there's so much more to data analysis. And so today I'm going to walk you through all of the steps, and including my thought process in those steps. And I'm going to use a real example. These are data that I am actually currently analyzing. Since these are unpublished data, I'm just going to share a subset of the data today so as not to give away the whole story. But I'll tell you that what I am seeing in this subset is pretty similar to what I'm seeing in the larger data set. I want to thank the PI of the study, Dr. Emily Krause, for allowing me to use her data in teaching. So this study is a cross-sectional survey that was administered online, and today we're going to look at data on 409 competitive adult female runners. And the study was interested in a syndrome called female athlete triad. So this is where athletes don't intake enough calories for the amount that they are expending in exercise. That can lead to energy deficiency, which can in turn cause women to lose their menstrual periods and have lowered levels of estrogen, which in turn can cause them to lose bone and have fragile bones. So the research question for this study was, do runners with female athlete triad have an increased prevalence of depression or anxiety? The main independent variable for this study was something called the female athlete triad risk score. So this is a validated risk score that's actually used to screen athletes for female athlete triad. And you get points for low energy availability, which is related to restrictive eating behaviors, for low BMI, for irregular periods, for late menarche, which is a delayed first period, and for having a history of bone stress injuries. The dependent variables are depression and anxiety, and these were measured by a validated questionnaire called the PROMIS questionnaire, which is actually frequently used in research. All right, so now let's talk about the steps of a data analysis. I've broken it down today into 5 main steps. So you're first going to process and clean your data to get it ready for analysis. Then you need to spend time familiarizing yourself with all of the variables in your data set. This is going to help inform step 3, which is when you actually get to test the main hypotheses of your study. Then you need to check for robustness. So what this means is you're going to make small tweaks to the analysis and make sure that the whole thing doesn't fall apart. If it falls apart, then it's not robust. And finally, you have to get everything ready for publication, which includes coming up with a story in a set of tables and figures that tell the story of your data. You spend some time really thinking about how to put those together so it's a clear story. And you have to prepare final code documents. When I do a data analysis, I actually spend the majority of my time in steps 1 and 2, and they're time consuming. And I would also argue that these are the most important steps of any good data analysis. And this is because if there are quirks in your data that you are not aware of, or if there are errors in your data, it doesn't matter how good your statistical model is. It's a garbage in, garbage out situation then. And I actually think that a lot of the errors that I see in the published literature arise because researchers often neglect steps 1 and 2. Everybody wants to rush to step 3, to testing those main hypotheses. But if you don't do due diligence on your data, that really is a big source of errors. Again, unfortunately, I think everybody wants to rush to step 3, and most statistical tests or many statistical courses actually focus almost exclusively on step 3 there, on how to do specific statistical tests, like how to run a t-test or how to run an ANOVA. And that may give researchers the mistaken impression that data analysis is synonymous with step 3 up there, when in fact step 3 is just a tiny sliver of a well-done data analysis, as we're going to see today. So what I'm going to do now is I'm going to walk you through the steps of a data analysis using this example of female athlete triad and mental health. So first thing we're going to do is process and clean our data, and there's just a lot of things involved in this step. It does take quite a bit of time. And you might think, oh, this step is going to be really tedious, but I actually really enjoy cleaning data. So there's something fun. There's something that's a little bit of like numerical detective work when you're digging into your data and trying to find like all the things that could have gone wrong. So I find that fun. And then there's just something really satisfying about cleaning your data. I think it's kind of like when you go into the back of that closet that you haven't cleaned for like 10 years, and you pull everything out and you kind of wash down the shelves and throw things out and organize everything. There's just something really nice about knowing at the back of the closet, even though you can't see it, that it's in order. And I feel the same way about just knowing that my data are in order. So the first thing you're going to do in this step is you're usually getting data in from somewhere like Qualtrics or REDCap. And what you're going to do is you're going to immediately import those data into a proper statistical analysis program, something like R or SAS, where you can write code to do your data processing and also your analysis eventually. And I want to make the point that you really shouldn't be processing data in Excel. And the reason I bring this up is that I've realized over the years that there's a lot of researchers who are very comfortable in Excel, and so they're tempted to do a lot of their data processing in Excel. This is just a bad idea because it's highly inefficient. I'll give you an example. I got brought in on a project a few years ago, and when the data were entered in REDCap, there had been some issue where I think some of the baseline data points had been entered in a follow-up column. And so there was an error. The PI of that study had downloaded the data into Excel and was going through observation by observation to realign everything, which probably took her 30 or 40 hours of work. I was able to download the data from REDCap and dump it into SAS and write like 4 lines of code and fix the problem in 20 minutes. So if you're processing data in Excel, you're probably wasting time. If you could write some code to do the same thing, it's going to be much more efficient. I also don't like Excel for processing data because when you're working in Excel, there's a high chance of accidentally introducing errors, and there's no real record of them, and it's not reproducible. The other thing is when you update your data, say in REDCap, if you've done everything in Excel, you're going to have to download and you're going to have to repeat all of those manual steps in Excel. Whereas if you've written code, you just port your data, your new data back into SAS or R, and you rerun your code. It takes 2 seconds to update the data. So other things we're going to be doing here is I spend a lot of time renaming variables. So they come in from Qualtrics or REDCap with funny names, and I'm not going to remember those names. So I'm going to name them something I can remember and type easily. I'm also going to be calculating new values. So in this example data set, the participants took a questionnaire about depression, and it was 8 questions long. Those 8 questions get processed into a single depression score. So I'm going to write some code to do that. I'm also going to spend a lot of time rummaging around in my data checking for errors. And this is time consuming. And I actually spend a lot of time actually emailing back and forth with the PI and the people who are maintaining the database to see if we can resolve those errors. Or sometimes they're not errors, they're just weird data points, and we have to decide how to score those people. We're also going to worry about how to handle missing data, and I'm going to figure out, if any observations need to be excluded. All right, just for fun, what I'm showing you here is the data processing and cleaning code that I have written for this particular data analysis. This is SAS code. I do most of my data processing in SAS, actually. But you can see it's 12 pages long. So this is a lot of work, data processing and cleaning. In contrast, I'm going to show you a regression model later. That regression model took like 5 lines of code. So a lot of work goes into data processing and cleaning. So as I mentioned, we're going to get data. Often for research data is going to be something like from Qualtrics. The data for this study were entered into Qualtrics. So what I'm showing you in this screen now is what the spreadsheet looks like when I download the data from Qualtrics. And I'm just showing you the variables for height and weight. So height was entered actually in 2 separate columns. They asked the participants to report their height as feet, and then separately as the remaining inches. And then weight was just asked in 1 column as weight in pounds. Notice that the variable names here are really awful, like Q93 underscore 39 underscore text. I'm never going to remember that. So I'm going to rename that variable into something easy like height. I'm going to write some code that's going to clean this data up. I'm going to rename the height variable to height. I'm going to combine those 2 columns into a single column. So if you entered 5 feet 7 inches, I'm just going to calculate that you're 67 inches tall. Weight, I'm going to rename that variable. And then I calculated BMI from their height and weight. The height data actually makes a really good example to talk about checking, the need for checking your data for errors. So that variable was asked in sort of a weird way. They were asked to answer how many feet are you and then how many inches are you. So like you were supposed to put in 5 feet, 4 inches. But there were a lot of variations on how that got entered. When I went to calculate height, what I did was to take their feet variable, multiply by 12, and add inches to get the height in inches. But that led to some impossible and weird height values, which I then fished out of the data set to try to resolve what was going on. So for example, some people came out with heights of like 124 and 125 inches. Well, why did that happen? So actually, it turned out I could figure out some of what went on here. So some people, when they were asked to enter feet, they put 5 feet. But then when they were asked for the inches, they didn't put the remaining inches, like 4 inches. They put their height in total inches, like I'm 64 inches tall. Well, obviously, when I went to calculate height, I came up with this weird value. But this one is resolvable. I know what they meant. They meant that they're 64 inches tall. So I can easily fix that. That is not a problem. I'll just change that to 64 or 65 inches. Another thing people did is when they were asked the height, they put in, for the feet, a decimal place, like I'm 5.44 feet tall. And then they didn't enter anything for inches. So when I went to calculate height, I got a missing value. But this one is also resolvable, right? I know what they meant. I'm just going to take the 5.44 and multiply by 12. And I am able to fill in, for example, that 5.44 feet corresponds to 65 inches. No problem. Now, there were some heights that in the end I couldn't resolve. They just had weird values. So, somebody entered 8 feet, 8 inches. Maybe they meant 5 feet, 8 inches, but I can't really tell. Somebody entered 2 feet, 4 inches. I don't know what that means. So these heights actually can't be calculated, which means I'm not going to be able to get a BMI for this person. So I'm just going to leave those data as missing, and these people will be missing a BMI. Just fun little story, though, the magic of crowdsourcing on Twitter here. Last week, I posted on Twitter about this upcoming webinar, and I showed this particular slide. And an observant researcher out there, Kat Daniels, tweeted back and said, hey, that one that says 1 foot, 85 inches, maybe they used metric units. I hadn't thought of that, but it totally makes sense, right? 1 meter, 85 centimeters. That translates to about 72 or 73 inches. So we may have resolved that one data point just through the magic of Twitter. All right, another thing we had to do with these data is the participants took this PROMIS depression questionnaire. And this consists of 8 questions about depression. And they're things like, in the past 7 days, how often have you been bothered by the following symptoms? And it's things like, I felt worthless or I felt sad. The participants answer this question on a Likert scale, rarely, never, sometimes, often. And those answers get translated, all 8 answers on those 8 questions get translated into a single depression score that ranges from 8 to 40. So I wrote some code to process those data, and I named the new variable dep now for like current depression, and we end up with a score between 8 and 40. Now, for this particular variable, I did have to think carefully about missing data. So you'll notice that some people didn't fill out any of the questions. These are actually pretty easy cases because they're just going to get a missing value for depression. In fact, what happened on this, because it was an online survey, is that some people signed up to take the survey. They got like 2 or 3 questions in, and then they abandoned the whole thing. They decided maybe it wasn't worth their time. So we have a bunch of people who basically have blanks on everything. Obviously, they're going to be excluded from the analysis in the end. So those cases are in some ways easy. But then I have cases like the one that you see up here now, where this person entered 7 of the 8 questions, but they neglected to answer question 6. So what do I do with this person? Do I give them a depression score, or do I treat their depression score as missing? I have to think carefully about what to do here. And so to figure that out, what I did was to do a little bit more detective work. Is I went into the data set and tried to quantify how often were people missing items on the depression questionnaire. And so I just counted up how often were people missing 0 items, meaning that they answered all the questions. How often were they missing 1 item? They just missed 1 question. How often were they missing all 8? And fortunately, it turns out that most of the cases are pretty easy. Most people either answered all of the questions or they answered none of them. So those are easy cases. We only had 5 participants who had partial data on the depression questions. So there were 3 people who actually missed 1 item. There was 1 person who missed 3 items, and there was 1 person who missed 4 items. So I had to make just a decision about what to do about these people. And so it made sense to me to say, hey, if you're missing 1 item, I can just give you the minimum score on that 1 item, and I can still give you a depression score that's going to be reasonably accurate. But if you were missing 3 or 4 items, that seemed like I wasn't going to be able to get an accurate depression score. So I'm going to count those people as having a missing value for depression. So overall, I ended up with 58 participants who didn't get a depression score. And in this particular analysis, there were 3 main variables: the triad risk score, the depression score, and the anxiety score. And if you were missing any one of those, you really couldn't contribute much to the analysis, since those were the key variables. So when we started here with 476 people who started the survey, but when I excluded people who were missing any one of those 3 main variables, we ended up with a final sample size of 409. All right, so what are we going to do next? Now we're going to get to step 2. This is a step where you familiarize yourself with all the variables in your data set. You really have to dig in to make sure that you understand all of the variables and really understand what's going on in your data. This is really going to help inform step 3, the model building step. So what are we doing here? Well, one thing I'd love to do is I'd love to make a bunch of histograms with my data. I just plot a histogram of each individual variable to look at the distribution. And actually, fun story, I teach a 3-quarter statistics sequence at Stanford. So I have many of the same students all year long, and I've probably been presenting a lot of histograms to them all year long. So in the spring quarter, we do these data analysis projects. And so the other day, they had just gotten a new data set, and they were working together in teams, in breakout rooms on Zoom, and I was floating around the breakout rooms. And my heart was warmed so much because immediately, a lot of my students were immediately drawing histograms of their data. So it was their instinct to look at the distributions in histograms. So I felt like I taught them something. I'm also going to be, in this step, calculating descriptive statistics. I'm going to be really understanding each variable, and I'm going to be exploring simple relationships between the variables. Which variables go together? So as an example, here's the histogram for age in my data set. So what are we looking at? A histogram. On the x-axis, we have different ranges of age. On the y-axis, we have how frequently different ages occurred. So for example, I had, this is roughly about 18 to just over 20. I had about 10% of my sample in that 18 to 20 bucket. What turned out to be interesting here is that I actually have a pretty wide range of ages. So I actually have some women who are in their late 50s, and I have some women who are actually postmenopausal. And this turns out to be really important. I had to email back and forth with the PI of the study to say, hey, how do we score postmenopausal women for menstrual irregularity, which is one of our key variables? Just being aware that the age range is so wide also reminds me that I'm going to need to control for age in my analyses. This next histogram that I'm showing you shows the distribution of the triad risk scores. Remember, this is my main independent variable. So this risk score ranges from 0 to 10. And 10 means that you have more traits of the female athlete triad. I'll just point out that we had only one person who scored a 9 and only one person who scored a 10. So those really high scores were actually rare. I'm going to say a little bit more about where this score comes from now. So the triad risk score comes from summing up 5 individual components that are each scored on a scale from 0 to 2. So for example, you get points for menstrual irregularity. If you have normal periods, your score is 0. If you have somewhat irregular periods, you get a score of 1. And if you're missing periods altogether, you get a score of 2. You also get points for late menarche, a late first period. So if you had your first period at age 15, you get a score of 1. If it's age 16 or older, you get a score of 2. You get points for prior bone stress injuries. So if you've had one stress reaction or stress fracture, you get 1 point. If you've had more than one, you get 2 points. You get points for a low BMI. So under 18.5, that's 1 point. Under 17.5, that's 2 points. And then finally, you get points for low energy availability. And this is scored actually in this data set based on a series of questions that we asked the participants about their eating behaviors. And we were really looking for restrictive eating behaviors that might limit the amount of calories that somebody was taking in. I think this low energy availability risk score actually turns out to be kind of an important part of the whole story here. So I'm going to say just a little bit more about this risk score. So the participants were actually asked a pretty long series of questions about eating behaviors, including, a history of eating disorders. Had they ever been diagnosed with an eating disorder? And they were asked if they engaged in certain restrictive eating behaviors, and they were asked how frequently they engaged in those behaviors, and also things like purging. If they had a history of eating disorders or a frequent restrictive eating behaviors, those things would give you a high score on the low energy availability risk score. And I'm just showing you here the histogram for that variable, the history of an eating disorder. I think this variable is kind of interesting and actually an important part of the story here. So this is a binary variable. We asked women if they'd ever had an eating disorder diagnosed. You got a 1 if you did and a 0 if you didn't. And I thought what's interesting here is actually the rate of eating disorders, or at least a history of eating disorders, was actually pretty high. It was about 30% of the sample. So that struck me as kind of high. Now, to be fair, it was kind of a broad group of eating disorders. It included subclinical eating disorders like disordered eating. So we may have seen a high rate just because it was kind of a broad definition of eating disorder. But it also does make me worry a little bit that we're going to have to worry about selection bias in this data set, right? The people who filled out this questionnaire might have been, people who had these issues might have been more likely to fill out the questionnaire because they were interested in these issues. So that's something we're going to tuck in the back of our minds. All right, the main dependent variable here is this depression score. This is the PROMIS depression score. And I'm showing you the histogram here. It ranges from a score of 8 to a score of 40, higher scores being more depression. We have a pretty wide range of depression scores in this data set. We also measured the PROMIS anxiety scores. Those range from 7 to 35. Again, we have a pretty wide range in the data set. Besides drawing a lot of histograms, another thing that I'm going to do is to run some quick correlation matrices in my data. All this does is give me a quick sense of which variables go together. These are not correlation coefficients that I'm going to report in the paper. I don't care about the p-values. This is just to give me a really quick sense of which variables are related with one another. And by the way, the correlation coefficients that I'm showing you here are Pearson correlation coefficients. And you might wonder, like, but some of these variables are not normally distributed. In fact, if I calculated the Spearman correlation coefficients here, I'm going to get basically the same thing because we have a pretty big data set. We have over 400 people. So it actually doesn't really matter which type I calculate. And again, I'm not going to be reporting these in the paper. This is just to give us a sense of what goes together. So for the first thing that pops out when I did a correlation matrix for depression and anxiety is that depression and anxiety are correlated with one another. Now, that's not too surprising, but it's important for me to recognize because whatever story I see for depression, it's likely I'm going to see a similar story for anxiety. And actually, for just sake of time, for the rest of today's talk, I'm really going to focus just on the depression score. Another interesting thing that jumped out is that of all the triad risk factors, the one that really jumped out as being the most correlated with depression and anxiety was this low energy availability risk score. So I actually think this is going to be an important part of the story here. Overall triad risk score was also modestly correlated, but a little bit less so than that low energy availability score. Another thing that popped out that I thought was interesting is this eating disorder. If you ever had an eating disorder variable popped up as having some correlation with depression and anxiety. And this is actually not surprising because we actually know from previous research that eating disorders tend to co-occur with depression and anxiety. But I think, again, this is going to be an important part of the story of our data, so it's important to recognize. Another thing that jumped out is that there is a slight inverse correlation between depression and anxiety and age. The correlations are negative. Older women had slightly less depression and anxiety. That's just going to be important to put in the back of our minds to remind us that we're going to need to adjust for age. All right, the last thing that I want to note on this PROMIS depression score is that the PROMIS depression score can be broken down into categories. The people who put out this tool have a set of ways of breaking this up into categories that's also been validated. And so you can break it up into none, mild, moderate, and severe depression based on their underlying score. And so for this analysis, we actually decided a priori that we were going to look at the depression groups rather than the underlying score because it had a little bit more clinical relevance to look at the depression groups. What I'm showing you next are some descriptive statistics for those depression groups. And so here are the sample sizes of those 4 groups. And what's interesting to note is that we had a lot of people with mild or moderate depression, but that severe depression group was actually pretty small. I also calculated the average triad risk score for each of these groups. For no depression and mild depression, it was 2.4. So it actually didn't go up in the mild depression group, but it did go up to 3.4 in the moderate depression group and up to 4.9 in the severe depression group. And then the other thing that I found interesting, again, this eating disorder variable keeps popping up. So if you look at how many people had a history of eating disorders across these depression groups, it has this nice progression where it starts at 23% in the no depression group, goes up to 30%, then 41%, and then a whopping 69% in the severe depression group. So again, I think the eating disorders are a part of the story here. All right, so that brings us now to our third step when we're actually going to be testing the main hypotheses of this study. So what kinds of things are we going to be doing in this step? So first of all, we're going to follow our analytic plan. You want to have some analytic plan before you start. You want to test assumptions of the model, and you want to do a lot of that testing just by visualizing the relationships that you're trying to test. And I think one of the common errors that I see when I'm reviewing papers is often authors will present models, and I'm pretty sure that they never visualize the relationships behind those models. And that's a really big problem because you're not fully testing the assumptions of the models unless you've actually looked at the data visually, graphically. And also, sometimes you're picking up on artifacts. If you haven't looked at the data visually, you may not realize that. So we're going to build final models, and we're going to interpret the results. For this particular example, as I mentioned, we decided we were going to look at the depression groups, this none, mild, moderate, and severe depression, rather than an underlying continuous score. So that meant that to test the association between triad risk scores and depression group, we had to use either ordinal or multinomial logistic regression. We couldn't use binary logistic regression here because our groups, we have more than 2 groups, we have 4 groups. I'm going to show you just the results today from the multinomial logistic regression, just for time's sake. I'll tell you that I got a slightly better fit with the multinomial regression than with the ordinal regression. And I've adjusted these models for age because, of course, that's a potential confounder here. The other thing we wanted to do was to explore our data to see if any one of those components of the triad risk score seemed to be more important than the others. So now I'm just reminding you, here's what the promised depression groups look like. And I've color-coded them today. So here's none. And then for the mild group, that I've color-coded in purple. Moderate is colored in yellow, and severe is colored in green. And I'm going to stick with that color theme for the rest of today. So one of the first things I wanted to do was just to think about, how do I visualize the relationship between these depression groups and the triad risk score? And I wanted to come up with something intuitive to start. So I'm showing you that graphic now. So what you're looking at is, remember, the triad risk score is an integer score. It's 0, 1, 2, 3, all the way up to 10. There were only 2 people who had scores of 9 and 10, though. So I grouped 8, 9, and 10 together because I wanted enough people to be able to calculate the percent with each of these types of depression. So for example, if I look at everybody who has a triad risk score of 0, that's this group down here, what I've got on the y-axis is, this is the percent with each type of depression. So for example, if you had a triad risk score of 0, about 28% of that group had mild depression. An additional about 10%, that's the yellow bar here, had moderate depression. And then the green bar here, that's just a sliver, about 1% had severe depression. And so this makes kind of an intuitive visualization of the relationship. And you can see there is a trend here where it does seem that the depression goes up as you increase in the triad risk score. And in particular, you'll notice that there's a lot more green at those high triad risk scores. So it seems like the severe depression really goes up. And to make this a little bit easier to see, I'm now going to separate out for you on the right-hand side here, mild, moderate, and severe depression. So if I just look at the percent with mild depression at each one of these triad risk scores, what you'll notice is actually it's pretty flat. Mild depression does not increase with increasing triad risk score. But if you look at moderate depression does seem to go up, although it plateaus a little bit after about a score of 5. And then finally, if you look at severe depression, there really does seem to be a pretty steep increase in severe depression as you get to those higher triad risk scores. So I think this particular graphic is a nice intuitive visualization of the relationship. The graph that I'm going to show you on the next slide is very much related to this graph, but it's just slightly more complicated. So the graph I'm going to show you next, I'm going to graph not the percent with depression against triad risk score, but I'm going to graph something called the logit of depression against triad risk score. Just keep in mind that the logit is just a mathematical transformation of the percentage. And so if the percent is higher, the logit will also be higher. So you'll see that it's the same general pattern. The reason that I'm bothering to do this more complicated graph with the logits is that because logits have nicer mathematical properties than percentages. So we take the percentage and we do this mathematical transformation. We come up with the logit, which has these nicer mathematical properties. So for example, a percentage has to range from 0 to 1. A logit can range from negative infinity to positive infinity. But I want you to just, what I'm showing you here is a scatter plot of our observed data. I'm plotting, again, this transformation of the percentage against the triad risk score. The pattern, though, is exactly the same as I showed you on the last graph. So these dots represent the observed data. And then what I've done is I've superimposed what's called a Loess line or a smoothing line on the data. And this is just to give me a sense of the pattern of the relationship. Ultimately, we're going to be fitting straight lines, as I'm going to show you. So we want to make sure that a straight line fit is a reasonable fit to these data. And indeed looks like straight lines would be reasonable. You look just like at the mild, the purple line here, you'll notice that just as before, it's pretty flat. No increase with triad risk score. The moderate depression goes up moderately, and the severe depression goes up more steeply. So what I then did is I fit a multinomial logistic regression to these data. All that means is that I fit 3 lines. And so here's the fitted model on the right here. It's just I fit 3 straight lines to these data. And you can notice that the fitted model looks pretty close to the observed model. So it's a pretty good, we're able to get a pretty good fit on the data. And what you see here is that the purple line, that's the mild depression, is basically pretty flat. So no increase in mild depression with increasing triad risk score. The slope of the yellow line, the moderate depression line, is kind of moderate. And so there's some increase in moderate depression as you go up in triad risk score. And then the slope for the green line is much steeper. So big increase in severe depression as you go up in triad risk score. Now, when we go to report these results for our readers, it turns out that most people can't think in logits. It's not an intuitive unit. And so these slopes, the slopes of these lines are informative, but they're in logit units, which nobody can understand. So there's 2 ways we typically present the data from logistic regression. So one thing we can do is we can take the logit and we can transform it back to a percentage, or what we call a probability, a predicted probability. So I'll show you that in a minute. The other thing is that these slopes actually have a direct translation as odds ratios, so we can also present odds ratios. So what I'm showing you now is, this is the same graph I showed you before on the left. This is just the observed percentages or observed probabilities that I showed you before. What we can get out of the logistic regression model, one of the things the model gives us is predicted percentages or predicted probabilities. And so that's what you're looking at here on the right. And of course, you'll notice that it's much smoother. It has this nice smooth pattern. Well, that's because we fit a model to the data, so everything is going to follow a nice pattern. But you can see it's kind of a reasonable pattern for given the observed data. And you can go in and you can look at, well, what would the predicted percentage with severe depression be for people with a triad risk score of 0 or for people with a triad risk score of 1? So you can report that in your paper. You can also report odds ratios. Logistic regression gives you odds ratios. And I think a lot of people are familiar with odds ratios, so I think this is a useful translation of the model. So what you're looking at here is the multinomial regression is going to give you odds ratios for the 3 different groups, the 3 different outcomes: mild, moderate, and severe depression. These odds ratios, the reference group is no depression, and the units are 1 point of triad risk score. So I'm showing you here, for example, for mild depression, the odds ratio is basically 1. Remember that for odds ratios, an odds ratio of 1 means no association. We already saw that, right? The triad risk score doesn't appear associated with mild depression. In contrast, for moderate depression, we do get a significantly elevated odds ratio. So how would you interpret this 1.29? So what this means is, and this is adjusted for age, is that for every 1 point higher triad risk score, every point you go up, that's associated with a 29% increase in the odds of having moderate depression relative to no depression. And then for severe depression, of course, you can see the odds ratio is even bigger. The other thing I wanted to tease out was I wanted to look not just at the overall triad risk score, but I wanted to look at those individual components of the triad risk score and see if any one of them dominated. So what I did next was I refit my logistic regression model, but rather than using the overall triad risk score, I entered those 5 separate components that each range from 0 to 2. So the low energy availability, menstrual irregularity score, low BMI score, late menarche, and bone stress injury scores. And these are each going to be adjusted for each other because, of course, they're correlated. And I wanted to see just if any one of them sort of jumped out as dominating. And so what I'm showing you now, these are the odds ratios from the regression model for severe depression. So on the x-axis, these are odds ratios. This vertical line that you see here, that's an odds ratio of 1, meaning no association. And what really jumps out here is there's a strong signal for this low energy availability score. Of course, the odds ratio is close to 5, but of course, the confidence intervals are really wide because remember, we don't have that many people with severe depression. So we see a strong signal for low energy availability and then some signal for menstrual irregularity and low BMI. But really, it's that low energy availability that seems to jump out. we look at the odds ratios for moderate depression, really after adjusting each of these for each other, it's really the low energy availability risk score that jumps out as having an elevated association with moderate depression. And once you adjust for that, the other ones don't seem terribly related to moderate depression. And so I actually think this makes sense. And I think what we're seeing, what's going on here in these data is, remember that low energy availability risk score is based on eating disorders and abnormal eating behavior, which of course have a psychological component to them. So I think what we might be picking up on is that for some women, part of the female athlete triad can involve eating disorders and abnormal eating behaviors. And we know that those things co-occur with depression and anxiety. So I think that's kind of what we're picking up here. I'll just share you the odds ratios for mild depression. They're all about 1. That's because we didn't see any signal with mild depression. All right, so the fourth step, what we're going to do next is we're going to check for robustness. And what do I mean by this? I mean that we're going to try to break our analysis. We're going to deliberately try to break our analysis. We want to make sure that our analysis doesn't find something just based on some quirk. We want to make sure it's robust. So we're going to try making slight changes to our analysis and making sure that it doesn't just all fall to dust. If it does, then it's not robust. So one thing you might be wondering about in this particular analysis is you might have noticed that I took that nice continuous depression score and I chopped it into categories. And you might have even been scolded by a statistician in the past for doing that, right? Where statisticians always say, oh no, you preserve everything as continuous. You don't want to lose information. So you might worry that what we're picking up on is some artifact of that categorization. So one thing I'm going to check, I'm going to go and just make sure that if I preserve depression score as continuous, that I still see a signal. So what I did is I took that continuous depression score and I ran a linear regression. The depression score is my outcome and the triad risk score and age is my predictor. And I just wanted to see if I got a signal. And I do still get a signal. The beta coefficient is 0.94, that's the slope. And all that means is that every 1 point higher triad risk score was associated with about a 1 point higher depression score. So I am still seeing a signal. Another thing that the PI especially was really worried about with this particular study is that the questionnaire was administered during COVID. And we all know that during the lockdowns on COVID, people were isolated, and that had an effect on mental health. So she was concerned that we might end up being just picking up on some artifact of the pandemic. So when she designed the questionnaire, she actually asked the participants to retrospectively answer questions about depression prior to COVID. So we not only had current depression groups, we also had pre-COVID depression groups. So I went ahead and reran my model on those pre-COVID depression groups just to make sure that I'm still getting a similar result. And I'm showing you the odds ratios here. These are similar to the odds ratios I got with current depression. So this is not just some artifact of the pandemic. There's other things we're going to need to worry about with this data set. So one of the things I might really worry about, of course, these data are all self-report. We didn't have people diagnosed by a physician. The depression and the female athlete triad, these are not diagnosed by a physician. This is self-report, so there's some room for error there. And then another thing that I'm really going to worry about with these data set is, as I showed you, we had a pretty high prevalence of eating disorders, and we also had a pretty high prevalence of depression. And this does make me worry about selection bias, right? People with these issues may have been more likely to fill out the questionnaire because they were interested in these issues. So we're going to have to worry about that when we're interpreting the data and we're writing up the paper. We're going to have to interpret that in context, realizing that there may have been some selection bias. All right, so the final thing we're going to do after we've finished all the other steps is we have to get everything ready for publication. And one of the main things that we're going to have to do here is we have to come up with a nice, clear, clean set of tables and figures that tell the story of our data. And this is a bit of a communication task as well as a coding task. The other thing, and of course you have to get those tables and figures all formatted correctly for the journal as well. That can take time. The other thing I'm going to be doing is I'm going to be taking my code, which I must admit, sometimes when I'm doing code, sort of for myself, it gets a little messy. I write little notes to myself that only I can understand. So I'm going to have to go back and clean up my code to make it readable for other people. So I'm going to take my code, which again might be a little messy when I'm just doing it quickly, and I'm going to go back and try to make reproducible code, which means I am going to add a lot of annotations that other people could understand. And I am going to make sure that I give enough code that somebody could reproduce all the statistics and graphics in the paper. You know, this task of going back and cleaning everything up can be a little tedious, but it's actually super useful, because when you have to go back over your code, sometimes you catch errors in that step. And so it's really good to have to go back and tie everything up and make it neat. You also need to think about, how am I going to put my data into tables and figures that communicates a clear story? I like this particular figure that I showed you earlier because I think it tells a pretty clear story. So I might actually decide to use this in the final paper. However, if you notice, when I prepared this figure, I did it for this talk today, and so I used kind of funky colors. I'm probably not going to keep green and purple in my figure for the journal. So I'm going to be cleaning this up a bit, like improving the color scheme. Maybe I'm going to make it shades of blue or shades of gray. When I'm making graphs for myself, I often don't bother to make them in high resolution. For the journal, I'm going to have to render everything in high resolution. I'm going to have to clean things up like the labels, and I'm going to have to increase the font size, so when you shrink everything down, it's readable. So this can take a fair amount of time to tidy up your graphics and make them good for publication. I'm going to point out one tool that I have found really useful over the years, in case you haven't come across this tool before. In preparing those final figures for publication, there's a tool called the PACE tool, P-A-C-E. It's put out by PLOS, but actually it's useful for submission to any journal. And it allows you to upload your figures, and it checks those figures to see if they meet journal requirements. And even though it's the journal requirements of PLOS, they're kind of the same throughout. And so it's going to check, do you have the right resolution? Do you have the right file type? Do you have the right size? And if you don't meet some of those requirements, it can sometimes even fix them for you. So I found this tool to be super useful when you get ready for submission to a journal. All right, just to recap here, and then I'll leave a little time for Q&A. Our steps of data analysis, we're going to spend a lot of time processing and cleaning our data and really getting to understand the variables in our data set. That's going to help us do a better job in testing the main hypotheses. And in that step, we're going to spend a lot of time visualizing relationships. Then we're going to make sure to check for robustness, make sure the whole thing is not just contingent on some tiny analytic decision. And then we're going to get everything ready for publication. Before we get to Q&A here, I'm just going to point out a few other resources. So I have written a statistics column for a journal for many years, for the journal PM&R. You can find a lot of topics there, statistical topics there. I try to write it in a clear and easy to understand way. And in fact, if I've made you curious about multinomial logistic regression, which I talked about today, I happen to have just written a column on that, and I'm posting the link here, but if you Google my name and multinomial logistic regression, it will come up, so you can learn more about that. And then I also teach the Medical Statistics Certificate Program for Stanford Online. And if you are planning to analyze your own data, this is a really good place to start, because I'm going to teach you some basic coding and also teach you how to look at and think about data and how to approach a data analysis. All right, I'm going to now pass this back to Jackie for Q&A.
We have a lot of questions that have been submitted today, so we'll get to as many as possible. Thank you all for submitting your questions. One of the questions that we've received is how learners can get more content from you. So we just covered that. So you can enroll in one of Kristin's courses to get more content from Kristin. Another question that has come up a lot is folks are interested in what program you recommend. It sounds like you typically use SAS, and they are interested in knowing if that is the program that you recommend. Do you recommend Python, R, or SAS, and why?
Yeah, that's a great question. I do a lot of my programming in SAS. I have to say that's a little bit of an artifact of my generation. That is, I learned SAS first. And so I literally can dream in SAS code. I do SAS so often, and I'm very, very fast in SAS. I picked up R afterwards, so I am not as fast in R. So I tend to default to SAS, particularly for data processing. But I often have to go back and forth between the two because some of the newest statistical methods and some of the better graphics are actually implemented in R. So I think there are advantages and disadvantages of both. SAS is still heavily used in public health. So if you're going to be in public health, you might have some reason to pick SAS. R is free, totally free, and the newer things tend to show up in R. So I'm happy to advise you on your individual situation if you want to email me. If you can learn a little of both, it's like knowing multiple languages, it's great. But my preference for SAS really is just an artifact of when I studied, and SAS was more dominant then. So I'll admit that's more my reason for using SAS as opposed to any logical reason.
Great, thank you. The next question is there a way to account for binary variables that may not be diagnosed? For example, a diagnosed history of eating disorders versus a history of eating disorders that may not have been diagnosed.
Yeah, so I didn't mention, just because of lack of time, that so the question that was asked in this particular data set, they were asked about both full-blown clinical, diagnoses like anorexia, as well as disordered eating. And then they were asked this whole series of questions that was actually trying to get at that if you were not diagnosed with an eating disorder, but you were frequently restricting your food, that could indicate maybe an undiagnosed eating disorder or the subclinical eating disorder. So we were definitely trying to get at that, and that's really reflected in that low energy availability risk score.
Great. And the next question is, if you are adding age as a covariant after seeing the correlation coefficients, does that not constitute a form of harking?
Okay, so that's a great question. So I am not adding age. It would be p-hacking or harking or whatever you want to call it. If I added age to the model and then I saw, oh, my p-value for the thing I care about, the triad risk score and the depression score, my p-value got better when I added age to the model. So I am adding age based on my knowledge that age is a potential confounder, and I'm not adding age or putting age in or taking it out based on how it affects the main relationship that I care about. We're really worried about p-hacking with regards to that main hypothesis. Am I doing things, fishing around to find a way to get a significant result in that main thing I care about? Whether age is significant or not, I don't really care. I'm only including it as a potential confounder.
Great. The next question is, wondering if you can differentiate between hypothesis testing in descriptive studies, means comparison, correlational studies, with explanatory studies where there is a need to have theory to explain the relation. In other words, is there any need to have theory to support comparison studies?
That's a great question. So one thing that often gets conflated in statistics is explanatory modeling, where we're testing specific hypotheses and we have some reason to do so, versus exploratory data analyses, where we're just describing the data. So those are two separate goals when you're, for example, building a regression model. You might build a regression model in both, but the goals are totally separate, and so the entire approach is different. What I showed you today was explanatory modeling. We wanted to test a particular hypothesis because we had reason to believe that association might exist. And all the analysis was focused on that association. It's totally valuable to sometimes do exploratory data analysis as long as you label it as such and are very clear that that's what you're doing. Because you might just want to, look at what associations are out there without any preconceived notions. Just make sure to label it that way.
The next question is, can logit transformation be applied to the probabilities in general to get nice data, or only when we use logistic regression in our analysis?
Yeah, that logit transformation is a typical transformation that's used in logistic regression. And so, again, it has some nice mathematical properties. It tends to get used in that context. It may have, mathematical transformations can have many uses. So that's the common one where you're going to see it, but doesn't mean it couldn't be used elsewhere.
Okay. And then why did you choose multinomial regression and not binomial or any other regression?
Great question. Yeah, so because we had 4 levels of that depression score, I didn't want to collapse it into like depression, no depression. Because why not use all of that information, the none, mild, moderate, and severe? So we were either going to use ordinal or multinomial. Both could be appropriate for these data. It turned out that, remember that mild depression doesn't really increase with triad risk score. So for that reason, the multinomial fit turned out a little bit better than the ordinal. And again, I'll refer you to my recent article on multinomial logistic regression if you want more details of that model.
Great. I think we have time for just a couple more questions. So the next question is, in dividing into depression groups, are the ranges predetermined, or do you have to decide on the binge ranges by yourself? And a follow-up to that was, if you have to decide on the bin range yourself, how would you determine the appropriate number of bins and their range?
That is a great question. Yeah, so the PROMIS depression score, it comes with predetermined bins that have been validated prior. So I'm not thinking about those ranges. Those come with the PROMIS questionnaire, fortunately, because you want those to be validated. You don't want them to be arbitrary. There are some cases where you might decide that there's a reason you want to take a continuous variable and divide it up into categories. Then you don't want to like divide it up to you don't want to look at your data and then decide what categories you're going to use, like based on the p-value. That's a form of p-hacking. You want to have some a priori reason. That's when you write your analytic plan of how you're going to divide up into categories. And so there's some thought that has to go in that if it's not something that's already validated. In this case, it was a validated set of categories.
Okay, and the next question is, can Likert scale data be analyzed using parametric tests if they appear normally distributed?
Sure. So in this case, we turned a bunch of Likert scales into more of a continuous score, right? Because we took 8 of them and we put them together. So we ended up with a bigger, more continuous score. But if you just had, like, if let's say we'd only asked one depression question and you had one Likert scale that was scored, say, 1 to 5, sometimes you're going to treat those as ordinal, and sometimes you're going to treat those as continuous. And if you have a big sample size, and it looks kind of, even if it doesn't look normally distributed, with a big enough sample size, it's actually okay to treat that as continuous values. But you'll see statisticians debate this back and forth, how should we treat those ordinal data? And depending on your end goal and what the data look like and how big the sample size is, you might make different decisions for different data.
Great. And I think we have time for one last question here. And that is, how can you know whether the statistical test you use on your data is the right one? And also with working with biological data where n is small, how can you determine distribution?
Okay, so the first one, how do I decide on the statistical model? It's usually based on what's the form of the dependent variable, and then also do I have correlated data? So there's a number of decision points that I'm going to look at in terms of what are the data, what's the appropriate statistical model. But then I'm, of course, going to check all those assumptions in the model, like that plot we were checking linearity, for example. So that's so there's often, for certain types of data, there's a pretty clear best model choice or a set of models. For biological data where you have a small n, you can draw a histogram, but if you have 5 people, you're not really going to be able to tell whether that's normally distributed or not. For really small data sets, you might be best off just reporting descriptive statistics or defaulting to non-parametric tests.
Excellent. Thank you, Kristin. We're getting close to the end of our webinar, so we'll need to wrap up. Again, thank you, Kristin, for this wonderful presentation. If you'd like to learn more from Kristin, we encourage you to join our medical statistics course. You'll have more than 20 hours of content in each course to learn from Kristin, as well as access to videos and exercises to sharpen your skills. Once again, thank you for joining us today, and we hope you have a great week, and we hope to see you again at our next session. Thank you, Kristin.
Thank you.